Assalamualaikum, this is BSM4501, Applied Plant Cell and Tissue Culture. We are going to look at anther culture experiment. What is anther culture? Anther culture is a technique by which the developing anthers at a precise and critical stage are excised aseptically from an open flower bud. They are cultured on a nutrient medium where the microspore within the cultured anther develop into callus tissue or embryos that will give rise to half-life plantlets either through organogenesis or embryogenesis. Hi, I'm Sofea. I will be your scientist in the future, inshallah. So, let me show you how to conduct this experiment of anther culture. First thing first, we need to have all of these materials. Anthers from pink aster lily, MS medium supplemented with different types of plant growth regulators, 70% ethanol, 2% sodium hypochlorite, sterile petri dish, scalpel or blade, forceps, and sterile distilled water. The most important step in tissue culture, we need to follow aseptic techniques and no cross-hand movement in laminar airflow. These are the procedures. Start with surface sterilization, rinse with distilled water, transfer to sterile petri dish, split the buds, remove the anther, remove the filament, place on culture medium, seal and incubate. Lastly, we need to examine the results of callus formation. Hi, first we need to find the flower bud from pink aster lily. So this is pink aster lily. Step 1. We have disinfectant, 2% sodium hypochlorite, 70% ethanol, sterile distilled water, seal, 2 sterile petri dish, stamens or anther from pink aster lily, flower buds of pink aster lily were collected. And let's enter the lab to start the anther culture experiment. We need to check all the materials needed and place in laminar airflow according to correct arrangement to prevent blocking of airflow. Step 2. Imagine that we add the laminar airflow. Disinfecting the benchwork area and also spray our hand with 70% alcohol. Now we are good to start the experiment. The flower buds are surface sterilized by fully immersing in 70% ethanol. Immerse for 60 seconds or 1 minute. %70% ethanol used as pre-treatment with other sterilizing agent and effective at killing microbes, also acts to prevent contamination. However, we need to flame the equipment every time we use them: forceps, scalpel, and flask. Flame sterilization to prevent the introduction of microbial contaminants to a sample. Step 3. The flower buds are surface sterilized by immersing in 2% sodium hypochlorite. The flower buds been immersed in the solution for 1 minute. Surface sterilization of explant is a process which involves the immersion of explants into appropriate concentration of chemical sterilants for a specific time, resulting in the establishment of contamination-free culture. Step 4. The flower buds were washed 4 or 5 times with sterile distilled water. This is the first rinse. We need to repeat the second until 5th rinse. Why we need to use distilled water? To remove the bleach or chemical solution by rinsing with sterile distilled water. Step 5. We have forceps and scalpel. The buds were transferred to a sterile petri dish and we take the stamen from the flower bud by split open using a scalpel. This is the stamen. Sterile petri dish is contamination-free, that being sterilized using autoclave to help prevent the contamination of new culture. Step 6. The anthers were removed without damage. The stamen is made up of 2 parts. It composes a long tube called filament and has a pollen-producing structure on the end. This oval structure called anther. Anther produces pollen while the filament holds the anther up. Step 7, followed by step 8. The filaments were removed. The anthers were placed horizontally on MS medium supplemented with different types of plant growth regulators. We have MS medium with auxin or cytokinin and gibberellin, also MS medium with auxin and cytokinin only. Plant growth regulators promote cell division, cell enlargement, flowering, fruiting, and seed formation. Step 9. We need to seal both petri dish of culture mediums. The petri plates were sealed. Now follow me to the incubator. Both culture medium been incubated in dark at 28 degrees Celsius. The requirement for both sterility and the avoidance of dehydration in plant tissue cultures can impose sealing requirements that severely limit the rate of gas exchange in and out of the culture vessel. Sealing with parafilm to protect tissue cultures from infection and desiccation is the most used method. Dark incubation is easier to regenerate plants after the callus phase as opposed to direct embryogenesis. Step 10. So let's check the result for callus formation. Is there any callus formation? The result obtained after 2 weeks of culture indicates that the embryos were emerging from anthers and showed the shoot's elongation. Why MS medium? MS medium is Murashige and scoop medium is a plant growth medium used in the laboratories for cultivation of plant cell culture. In this culture, MSACG medium was used for the development of microspore within the culture anther. The MSACG media contains the combination of MS medium and growth regulator of 2,4-dichlorophenolacetic acid 2,4-D as auxin and 6-benzylaminopurin as cytokinin, also gibberellins. The high concentration of auxin to cytokinin ratio helps increase the callus induction and give rise to embryos from the primary culture stage on. Furthermore, the presence of gibberellins in medium helps to promote shoots elongation. The medium that is used in another culture is MSAC media. This media is absence of gibberellins and contains MS medium added with 2,4-dichlorophenolacetic acid 2,4-D and 6-benzylaminopurin BAP. The higher concentration of auxin compared to cytokinin helps in generating embryos and promotes stem elongation. Here is the summary processes of anther culture on nutrient medium that forms callus and produces haploid plantlet. This is the result of haploid plantlet that been produced through anther culture techniques, pink Esther lily flower. Thank you.